Hi, so in this video I'm going to be talking about diffuse lymphatic tissue and lymphatic nodules and everything about the lymph nodes, tonsils, histology. So first of all, diffuse lymphatic tissue and lymphatic nodules guard the body against pathogenic substances and are the site of the initial immune response. That is clear. The alimentary canal, respiratory passages, the genital urinary tract are guarded by accumulations of lymphatic tissue that are not enclosed by a capsule. So that's what we mean by diffuse. It means that they are not enclosed by a capsule. And lymphocytes and other free cells of this tissue are found in the lamina propria. Lymphocytes and other free cells of this tissue are found in the lamina propria subepithelial tissue of these tracts. So here we will have the epithelial cells, and under them we have the lamina propria. So in the lamina propria we have connective tissue. So in this connective tissue we will have our lymphocytes and other free cells. As we know, in the lamina propria we have different types of cells. This form of lymphatic tissue is called diffuse lymphatic tissue or mucosa associated lymphatic tissue, MALT, because of its association with mucous membranes. So as we know that the mucous membranes are basically epithelial cells. So we have the epithelial cells in those areas of the body: the alimentary canal, the respiratory passages, the genitourinary tracts, etc., etc. So wherever there is mucosa and there is lymphatic tissue, that part of the body is acting as an immunity tissue. So it is called the mucosa associated lymphatic tissue. These cells are strategically located to intercept antigens and initiate an immune response. After contact with antigen, they travel to regional lymph nodes. So first of all, they are in the lamina propria, and then since, for example, we are in the respiratory passages, some antigens will travel through, let's say, through the nasal cavities, and then it will reach those parts of the tissue. They will go through the epithelial membranes, and then when they will reach the lamina propria, there they will get in contact with those lymphatic cells, and then from there, those cells, they will travel to regional lymph nodes where they undergo proliferation and differentiation. Progeny of these cells then return to the lamina propria as effector B and T lymphocytes. So after the differentiation, after the proliferation and then the differentiation, they go back to where they found the antigen in the lamina propria as effector B and T lymphocytes, basically to kill the antigen or to get rid of it if it's not a cell. Yes, so here in this photomicrograph of diffuse lymphatic tissue, this photomicrograph shows the diffuse lymphatic tissue in the lamina propria. So here we have the lamina propria of the large intestine. So here we are in the we are in the alimentary canal, more like, I don't know. The lower portion of two intestinal glands is also evident. Okay. The highly cellular diffuse lymphatic tissue includes fibroblasts, plasma cells, eosinophils. However, the most abundant cell component whose presence characterizes diffuse lymphatic tissue is the lymphocyte, which can be identified by a small, round, dark, stained nucleus. So all of these are lymphocytes, 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 lymphocytes. We have some eosinophils, we have some plasma cells, and we have some fibroblasts. But okay, so here we have intestinal gland, so it's going this way, it's going this way in the intestines, and then between them we have the lamina propria. Okay, we go to the next page. The importance of diffuse lymphatic tissue in protecting the body from antigens is indicated by two factors. So 1 and 2. The first one is the regular presence of large numbers of plasma cells, especially in the lamina propria of the gastrointestinal tract. Yeah, like the picture before, it's the gastrointestinal tract, a morphologic indication of local antibody secretion. So plasma cells, as we know, are differentiated B cells, or like effector B cells. They secrete antibodies. So here the importance of diffuse lymphatic tissue in protecting the body from antigens is indicated by two factors. The first one is the plasma cells that we find in the diffuse lymphatic tissue, and they secrete antibody that get diffused in the lamina propria. So whenever an antigen is present, those antibodies will opsonize it. The presence of large numbers of eosinophils. So we have plasma cells and we have eosinophils. So what will these eosinophils do? Also frequently observed in the lamina propria of the intestinal and respiratory tracts, an indication of chronic inflammation and hypersensitivity reactions. So when we find eosinophils in those areas of the body, it means that we have a chronic inflammation and hypersensitivity reactions, because as we know that eosinophils are the almost the main cells in the hypersensitivity reactions. And it is important to note that eosinophils are activated by interleukin 5. Okay, so to sum it up, the diffuse lymphatic tissue has two types of cells. We have the plasma cells for antibody secretion, and we have the eosinophils for chronic inflammation and hypersensitivity reactions. Lymphatic nodules are discrete concentrations of lymphocyte contained in a meshwork of reticular cells. Lymphatic nodules. Right now we're talking about something new, which is lymphatic nodules. So lymphatic nodules are discrete concentrations of lymphocytes contained in a meshwork of reticular cells. In addition to diffuse lymphatic tissue, localized concentrations of lymphocytes are commonly found in the wall of the alimentary canal, respiratory passages, and genital urinary tracts. These concentrations, called lymphatic nodules or lymphatic follicles, are sharply defined but not encapsulated. So they're sharply defined but not encapsulated. So the lymphatic nodules are not encapsulated. A lymphatic nodule consisting chiefly of small lymphocytes is called a primary nodule. However, most nodules are secondary nodules. The secondary nodules, they have a distinctive feature that include the germinal center, which is located in the central region of the nodule, and appears lightly stained in histologic sections. So the germinal center develops when a lymphocyte that has recognized an antigen in the periphery returns to a primary nodule and undergoes proliferation. The lighter staining is attributable to the large immature lymphocytes, lymphoblasts, and plasmablasts that it contains. These lymphocytes have large amounts of dispersed euchromatin in their nuclei. So basically, once a cell in the periphery recognizes an antigen, it comes and it comes to this area. For example, they're all darkly stained, and then after that, once it starts to proliferate, it kind of migrates to the central region, and this is the central region that is lighter because the cells in here have dispersed euchromatin, which means that the nuclei are active and they are proliferating and ready to go, rather than the dense heterochromatin of small lymphocytes. Follicular dendritic cells are also present in germinal centers, interdispersed between populations of B lymphocytes. The germinal center is a morphologic, and it's important to note that the follicular dendritic cells secrete interleukins that activate B lymphocytes, like interleukin 3. The germinal center is a morphologic indication of lymphatic tissue response to antigen. The presence of a germinal center represents a cascade of events that include activation and proliferation of lymphocytes, differentiation of plasma cells, and antibody production. Mitotic figures are frequently observed in the germinal center, reflecting the proliferation of new lymphocytes at this site. So basically, the lightly stained germinal center has different events. Let's give them numbers. So it presents a cascade of events that include activation and proliferation. They're just getting activated and they're allowed to proliferate. Differentiation, so they only get activated and proliferate for lymphocytes, but for plasma cells, they differentiate and antibody production. Mitotic figures are frequently observed in the germinal center, and antibody production, of course, from the plasma cells. Mitotic figures are frequently observed in the germinal center, reflecting the proliferation of new lymphocytes at this site. The number of follicular dendritic cells and macrophages in the germinal center often increases dramatically after a period of intense response to an antigen. Yes, so we are trying to present... Actually, follicular dendritic cells don't present any antigens, but macrophages do. So we will find them both in this area. As an intense response to an antigen. So for follicular dendritic cells, they're there to help differentiation of plasma cells. And we have a mantle zone, or corona, is present, and that represents an outer ring of small lymphocytes that encircle the germinal center. So we have the germinal center and we have the corona. The corona is darkly stained. And this one has small lymphocytes, so from here they will migrate to the germinal center to proliferate and differentiate for lymphocytes, to differentiate for plasma cells. Lymphatic nodules are usually found in structures associated with the alimentary canal, such as the tonsils, ileum, and vermiform appendix. Generally, nodules are dispersed singly in a random manner in the alimentary canal. However, some aggregations of nodules are found in specific locations. So aggregations of nodules give us tonsils. It gives us tonsils, it gives us Peyer's patch, and it gives us the vermiform appendix. Yeah, these are the things that come from aggregations of nodules. So we have three things really: one, tonsils; two, Peyer's patches; and vermiform appendix is at the end. Arises from the cecum. Tonsils form a ring of lymphatic tissue at the entrance of the oropharynx. The pharyngeal tonsils, adenoids, located in the roof of the pharynx. The palatine tonsils, or simply the tonsils, located on either side of the pharynx and between the palatopharyngeal and palatoglossal arches, and the lingual tonsils at the base of the tongue, all contain aggregates of lymphatic nodules. The palatine tonsils consist of dense accumulations of lymphatic tissue located in the mucous membrane. The squamous epithelium that forms the surface of the tonsil dips into the underlying connective tissue in numerous places, forming tonsillar crypts. The walls of these crypts usually possess numerous lymphatic nodules. The walls of these crypts usually possess numerous lymphatic nodules, like other aggregations of lymph nodules. Tonsils do not possess afferent lymphatic vessels. However, lymph drains from the lymphatic tissue of the tonsil via efferent lymphatic vessels. So it doesn't have afferent. So that is the difference between tonsils and nodes. Tonsils do not possess afferent lymphatic vessels. However, lymph drains from the lymphatic tissue of the tonsil via the efferent lymphatic vessels. Peyer's patches are located in the ileum, distal part of the small intestine, and consist of numerous aggregations of lymphatic nodules containing T and B lymphocytes. In addition, numerous isolated, single, solitary lymph nodules are located along both large and small intestines. Yes, so we have Peyer's patches in the distal part, or the distal portion of the small intestine, and we have just like solitary, single ones dispersed in the small and large intestines. And then we have the vermiform appendix. Arises from the cecum. The lamina propria is heavily infiltrated with lymphocytes and contains numerous lymphatic nodules. Although the appendix is often described as a vestigial organ, the abundant lymphatic tissue that it contains during early life suggests that it is functionally associated with bursa equivalent organs. With age, the amount of lymphatic tissue within the organ regresses, and it is difficult to recognize. Okay, that's it for today.